Back in the 1500s, Paracelsus, the father of toxicology, told us that everything is poisonous and that the only thing that differentiates a poison from a remedy is the dosage. I think I ate one too many berries. I'll be right back. Wow, those were some good berries. A little sour, but hey, it all comes out in the end. So today we're looking at pharmaceutical chaperones and titrating them to a therapeutic dose. And by that I mean one that's not going to be toxic and that's going to be high enough to work, but not too high and not too low. And I believe that I've seen data all over the place, whether it's a clinical trial, usually too high and too high and too high. And we've seen it in mice and also tissue culture. And I believe that we can look at the endoplasmic reticulum and the unfolded protein response and possibly some caspases that can help titrate chaperones to the therapeutic level. That's safe. Folding proteins correctly is extremely important, and that's what chaperones do. And pharmaceutical chaperones help the other chaperones do their job when they can't get it done. One thing that we want to do is we want a protein out there with the correct conformation, that's functional, that's hydroxylated and glycosylated correctly, so that proper cell signaling happens that can lower inflation. Inflation, yes. I think it's political. That can lower inflammation and lower apoptosis, which then also can excrete proteins into the basement membrane. And we're talking about collagen. And the whole video talks about collagen specifically. So it increases the thickness of the basement membrane and the strength of the basement membrane and increases cell proliferation. If you get too high a dose, it can fold that protein, but then it gets in the way of other enzymes, and the enzymes can't bind. And the chaperones that are supposed to be there can't bind to it because, say, for example, 4-PBA could be in the way. And if 4-PBA is in the way of heat shock protein 47, then you can have all kinds of problems because it's supposed to bring it through the endoplasmic reticulum. And as the pH changes in the Golgi apparatus, it lets it go and sends it on its way. But if you have too high a dose, that may not happen. It may get over-hydroxylated, over-glycosylated. When it does get out into the basement membrane, it can have different cell signaling. And it may not say, hey, we need more basement membrane here, or whatever needs to happen, whether it's cell proliferation, if it's broken, it may not be able to tell the cells around it what's going on. So we want it to be perfect. We don't want it to increase apoptosis or inflammation. And we want perfect cell proliferation. If something's damaged, we need cells to be made. If it's not damaged, we don't want cancer. So all these things are very important. And when we have a dose that tanks a chaperone that's supposed to be there, that's not a good sign. So we want to keep them normalized, where in a range where they should be. So when there's ER stress, it goes super high. And then when that stress comes back down, we want it to be normalized. We don't want it to be low. We don't want it to be high. And this video is made for any type of chaperone. However, there are certain slides that we're going to definitely look at the 4-PBA chaperone. At first, I thought 4-PBA should be the one to treat collagen 4A1, 4A2 mutation diseases. I'm starting to lean toward TUDCA or even a combination of both. But let's look at 4-PBA. It was first FDA approved to treat urea acid cycle disorders and at a very high dose. So, people are usually taking up to 20 grams of this a day, which is, can be 40 tablets. It's kind of crazy. But if it works for it, and it does, that's a good thing. So what else can it treat? In clinical trials, it's being used to treat ALS or Lou Gehrig's disease. And it may be acting as a histone deacetylase inhibitor, which can expose DNA so that that RNA can be transcribed from it and produce proteins that the cell needs. And it could be that there's certain mutations within the ALS spectrum that produce proteins that get caught in the endoplasmic reticulum and are unfolded and can be treated as a chaperone. So, with a chaperone. So 4-PBA would act as a chaperone at that point. The problem is they're not really using it at a low dose. They're using it more of a medium dose. And I don't know how that affects things. I do know that at first the FDA looked at their data and said, no, I don't really see that it made a difference. And then they re-looked at this, what seems to be the same data, and said, oh, okay, yeah, we can see, less than a 10% increase. And we have no other drug to treat this disease. So that's where they're at. I'm thinking that if they looked at it as a chaperone, we can definitely tell if it's working as a chaperone and what dose it needs to work at as a chaperone. Can 4-PBA, TUDCA, or any chaperone be titrated to a safe and therapeutic dose by UPR markers in the serum? And I say the serum because usually they're looking at the UPR markers in a biopsy or in a cell culture. So in this video, we're going to look at what unfolded proteins do to the endoplasmic reticulum, and in particular, collagen 4A1 and 4A2 heterotrimers, what happens when they agglutinate and all stick together in the endoplasmic reticulum. And to look at that, we're going to study the 3 branches of the UPR, and we'll see what happens when you give 4-PBA at a high dose and speculate what might happen at a low dose. And then we'll consider how to use GRP, or glucose regulated protein 78, which is also known as binding immunoglobulin protein. That is a chaperone that you can find in the endoplasmic reticulum. And we'll also look at CHOP, or CEBP homologous protein, which is an enzyme which is upregulated when apoptosis is increased. So can we use those two to find a therapeutic dose for chaperones? So the UPR is activated by unfolded protein aggregates in the endoplasmic reticulum or over-glycosylated proteins. Now, they can be over-glycosylated because they're staying in the endoplasmic reticulum too long. They're staying there because they're not folded and they're not making it out to the Golgi apparatus. So normally, the response by the UPR is that they want to eliminate these aggregates. They will either fold the proteins or they will send them to the proteasome to be degraded and recycled. And the effect of chronic stress on the ER is immense. It makes the patient not produce enough proteins, and especially not enough collagen 4A1, 4A1, 4A2 heterotrimer, which is in the basement membrane or extracellular matrix. And these markers of the UPR can leak out into the serum. And that can help us understand, is it causing apoptosis, or did we give enough of a chaperone that lowers the apoptosis and lowers some of these markers in the endoplasmic reticulum to a therapeutic dose. So a normal stressed cell has an endoplasmic reticulum that gets very big like this. And as it folds the proteins and eliminates the bad proteins, it relieves that stress, and the endoplasmic reticulum can shrink back down. It either does that or it sends it into apoptosis and the cell dies. But with collagen 4A1 and 4A2 disease, or Gold syndrome, what we see is that the cell stays with a chronically stressed endoplasmic reticulum that is enlarged. So this paper comes out of Dr. Tom van Agatmael's lab in the UK. He has a lot of journal articles on collagen diseases. Very interesting paper, this one is. Murray, kudos to you. It looks at a family where the father has a collagen 4A2 mutation and it is unaffected. The patient has the same mutation in collagen 4A2 and has had an ICH, an intracerebral hemorrhage. And when we look at this marker, it is protein disulfide isomerase, and it is an ER resident. It is there to stabilize the collagen molecule by making disulfide bonds in between the collagen 4A1, 4A1, and 4A2. So when they look at it in a patient, you can see that it's upregulated. That means the UPR markers in general are probably all upregulated, some more than others, of course. Then they want to know, is it co-localized together with collagen 4A2 that's mutated? In the father, they see a little bit of it. In the wild type, they're not seeing it. But in the patient, collagen is definitely stuck in the endoplasmic reticulum. In figure B, we're looking at the size of the endoplasmic reticulum. And you'll notice that the scale is exponential. So in the wild type, it looks like it's about 11. And in the patient, it's maybe 110, maybe 10 times as big. And the father is just slightly bigger than the wild type. So there's definitely some ER stress going on with the patient. Then we look at D, and this is a Western blot which is depicted below in E and F. So BIP, the chaperone that helps fold proteins, both of them, calnexin and BIP, help to fold proteins. BIP is elevated in the patient, not elevated in the father. Calnexin, the same. The unfolded protein response occurs when there's unfolded proteins in the endoplasmic reticulum. It can gum up that works and stop other proteins from being made. So it's very important that it recognizes it when there's over-hydroxylated and glycosylated proteins getting stuck in there. And what it will do, it'll increase the natural chaperones that occur in there that can fold that protein. And it could also decrease transcription of DNA to RNA or decrease translation of RNA to the protein. And if that does not work, the cell's either going to be in chronic stress or it's going to initiate apoptosis. So how does the UPR deal with all these unfolded proteins? All these aggregates getting in the way and jamming up the endoplasmic reticulum? Well, the first thing that it needs to do is to lower the global rate of protein production. Then it needs to expand the ER capacity and its volume so it can handle production of more protein. It will also induce luminal chaperones inside so that there's more folding capacity there, because that's the problem. It's not folding things. Also, it will increase endoplasmic reticulum associated degradation machinery. So this machinery sends unfolded proteins to the proteasome. The proteasome takes it and chops it up into amino acids and glucose and anything that it can recycle and reuse. There are 3 stress sensors inside of the ER lumen, which represent the 3 branches of the unfolded protein response. The first one is IRE1, or inositol requiring enzyme. The second one is PERK, protein ER kinase. And the third is ATF6, or cyclic AMP dependent transcription factor 6. They all send very unique signals out into the cytoplasm and upregulating targets that will alleviate ER stress. BIP is the most abundant ER chaperone, and it is called a master regulator of all the branches because it sits on the sensors. And when it comes off of the sensor, it comes off to fold unfolded proteins. When it does that, it activates the pathway. So if you find XBP1 upregulated, you know that it came from the IRE pathway, or ATF4 came from PERK, etc. All these upregulated target genes respond to cell stress, reactive oxygen species, and protein folding. And also they increase ER-associated degradation. So spliced XBP1 can degrade messenger RNA so that it cannot be translated into proteins. And the PERK pathway can negatively regulate translation of messenger RNA. And an introduction to collagen chaperones would not be complete without talking about heat shock protein 47. It is the only chaperone that specifically reacts with collagen proteins. It's very highly conserved throughout evolution. And that means you can find it in C. elegans and fruit flies, in mice and humans, and the sequence is very similar, which leads me to believe that it probably has a very deep importance that we haven't even discovered yet. It recognizes glycine X-arginine in the glycine XY sequence, which I've spoken about in previous videos. It interacts only with folded proteins, not the unfolded collagen, and it binds it in certain areas and blocks over modification or modifications from happening. So it doesn't have over-hydroxylation and glycosylation. And this is important for the protein, for cell signaling and for the transport of collagen through the endoplasmic reticulum and into the Golgi apparatus and out into the extracellular matrix. It is not increased with upregulation of the unfolded protein response, and it regulates IRE by displacing a BIP. So we're going to look at it up here. Here's our IRE1 alpha sensor. BIP normally sits on it, and it comes off to help fold proteins. When it does, heat shock protein 47 comes down and it sits on it. And when it does that, it can dimerize with another IRE, which sets off the cascade signaling. And when the BIP is free again, it will come back and sit back down, and this will be free again to keep BIP there. So many other people have considered looking at various components of the UPR as a way to treat chronic endoplasmic reticulum stress. Let's go back to the Murray paper, where he adds 4-PBA at a 10 millimolar in vitro culture to fibroblasts. First, we look at the wild type, and this is looking at protein disulfide isomerase, which we talked about before. Of course, you don't really see it in the father or the patient with PBA, but without PBA, it's there. But once you add it, because there's not a lot of collagen getting stuck in the endoplasmic reticulum, it gets downregulated. And compared to not having 4-PBA, there is a lot of collagen 4A2, along with the collagen 4A1, stuck into endoplasmic reticulum. Next, we look at markers in the endoplasmic reticulum. And these markers are also going down. If we look at calnexin, it starts out pretty high and is brought down, along with ATF4, which is in the PERK pathway. And it's brought down a lot, but it's still there. And here is BIP. Now keep in mind, BIP is the most abundant chaperone in the endoplasmic reticulum. And it takes it from a 3 to maybe a 0.1. Now, why I say that we can use these markers to adjust dosage is because this is not what I want to see as far as a culture. Now, keep in mind that a cell culture is way different from in vivo, whether a mouse or a human. Things don't always express the same. But if they were to redo the experiment, they would probably want to bring BIP within a range of what is wild type or the father, and not bring it down here so far. So they might want to go with a dose that is less than 10 millimolar next time. And keep in mind, this is a very intensive paper. They looked at all kinds of stuff, studies with vitamin C, whether the proteins went to the proteasome or autophagy, and it was a very ambitious paper. It was not something that looked at adjusting a dosage even in vitro of 4-PBA. So next, when we think about the amount of 4-PBA, we want to remember that the size of the endoplasmic reticulum can go up to 9 times the size when you have a lot of ER stress. So this is our endoplasmic reticulum. This is the part that would probably get much bigger. It is the rough endoplasmic reticulum. The ribosomes translate messenger RNA. The smooth endoplasmic reticulum is primarily for fatty acids and hormones that are produced. We know that BIP can be found in each branch of the unfolded protein response. In a moment, I will show you that it can be found in the serum. But what other markers can we find in the serum to help us adjust chaperone treatment levels? Back to the Murray paper. Let's look at apoptosis when 4-PBA and absorbic acid are administered. Now keep in mind, absorbic acid is the main component of vitamin C, and in the wild type, and this is fibroblast culture, when it's given, it reacts very well. There's less cell death. You add the 4-PBA, there's less cell death. That's what we expect. A very curious thing happens when we look at the patient. Of course, there's more apoptosis going on because the collagen 4A2 is mutated and is getting stuck in the endoplasmic reticulum, increasing stress. And when they add absorbic acid, cell death goes up. Very strange, very strange. So it begs the question, why did that happen? Well, I need a short video to explain that to you. But keep in mind that absorbic acid, vitamin C, is necessary to have hydroxylation of collagen proteins. Without it, you have scurvy, and you really can't live without vitamin C. So then they add the 4-PBA, and it goes down. So it would really be cool if we had some type of marker that when the ER stress got high enough and it set off a marker that you could see that would lead to apoptosis, that you could measure it in the serum. So Liu Z et al. Provides us with this diagram of the UPR mechanism. It has all 3 of our UPR branches. You'll note that the ATF6 and IRE1 both come down to activate CHOP, which can increase apoptosis. The perk mechanism can also produce ATF4, which increases CHOP, which increases apoptosis, but it also has another cascade that leads to various caspases, and they can increase apoptosis. There's a few caspases that seem to inhibit apoptosis, but let's talk about CHOP and why it can be found in the serum. CEBP homologous protein, aka CHOP, is a member of the CCAA T enhancer binding protein family of DNA binding transcription factors. What does that mean? There's a sequence. It's called a cat box, and it has that CCAT sequence in it. And it is usually 60 to 100 base pairs upstream of where the initial transcription factor binds to transcribe DNA into RNA. It is a pro-apoptotic transcription factor that is ubiquitously expressed throughout the cell, and you can also find it in the serum. And there's many mouse models that study this particular factor. And in the diabetic mouse model, they have insulin that is being expressed at a high level, and it is causing insulin resistance and inflammation throughout the mouse. But when you have a deleted gene that expresses CHOP, you have reduced oxidative stress, and the beta cells in the pancreas are less stressed, and they produce then a therapeutic amount of insulin. And it can be found right next to CHOP in initiating apoptosis. The main resident and master regulator, BIP, or GRP78, can be found in the serum, along with CEBP homologous protein, or CHOP, which mediates ER stress-induced apoptosis. Since both of these can be found in the serum, can we use them to find a therapeutic dose for our chaperone? And are there other markers that we can find in the serum that can help us do this too? So the next video is looking at how we can use the UPR to titrate a therapeutic chaperone dose that is just right. Like, subscribe, check me out on collagen and rarediseasefoundation.com. Thanks.